Here's a sample protocol for Southern hybridization. Materials. DNA samples to be analyzed. Agarose gel electrophoresis equipment. Transfer buffer, e.g., 10xSSC. Nitrocellulose or nylon membrane. UV crosslinker or oven for membrane fixation. Hybridization solution, e.g., DIG Easy HYB or similar. DIG labeled probe, or other labeled probe of choice. Washing buffer, e.g., 2xSSC plus 0.1% SDS. Procedure. Prepare DNA samples. Isolate and purify genomic DNA from the cells of interest. Digest the DNA with restriction enzymes if necessary, and run the digested DNA on an agarose gel. Transfer DNA to a membrane. After gel electrophoresis, depurinate and denature the DNA in the gel. Prepare the transfer buffer according to the manufacturer's instructions, and transfer the DNA from the gel to a nitrocellulose or nylon membrane. Crosslink the DNA to the membrane using a UV crosslinker or bake the membrane in an oven. Pre-hybridize the membrane. Prepare the hybridization solution according to the manufacturer's instructions, and preheat it to the appropriate temperature. Pre-hybridize the membrane in the hybridization solution at the recommended temperature for 1 to 2 hours to block nonspecific binding. Hybridize the membrane with the probe. Dilute the DIG-labeled probe in the hybridization solution, and add it to the pre-hybridized membrane. Incubate the membrane with the probe overnight at the recommended temperature. Wash the membrane. After hybridization, wash the membrane with the washing buffer at the recommended temperature and stringency to remove unbound probe and reduce background signal. Detect the signal. Use an appropriate detection system, e.g., chemiluminescence or colorimetry, to visualize the bound probe on the membrane. Analyze the results. Interpret the results to determine the presence and size of the DNA fragments of interest. Note: this protocol is a general guideline and may need to be optimized for specific samples or probes. Consult the manufacturer's instructions for detailed protocols and troubleshooting tips.